Hello, I'm Marla Tharp, and I'm a researcher at the Carnegie Institution for Science, Department of Embryology. In order for a species to survive and be very successful, they need to be able to adapt to environmental changes. And for this reason, our genomes have built in numerous mechanisms to maintain this potential to diversify and evolve in response to environmental challenges. And one really enigmatic way that they can do this is by keeping around a massive number of transposable elements or jumping genes in the genome. And indeed, our own genomes have about 50% transposable element DNA. And to put this in perspective, only about 1 to 2% of our genomes encode functional proteins. So I became very interested in transposable elements because they're a paradoxical genomic feature. They're important drivers of evolution. They've been kept around in our genome because they have the ability to try out new variations of genes and see if these novel phenotypic traits are advantageous, but also they are dangerous and they are selfish elements, and their goal is to propagate themselves throughout the host genome at the expense of the host even. And so this can create a lot of DNA damage and also pathological mutations that threaten host cell viability. And since these transposable elements can be so dangerous, most of the cell types in our bodies try very hard to keep them quiet. They use a number of repression mechanisms to prevent the expression of transposable elements, except for one cell type, and that's the developing oocyte or egg cell. And so here we're looking at a photo of a mouse ovary of embryonic day 18.5. And here you can see in green this high expression of line 1 retrotransposon. And line 1 is actually the only remaining active element in humans that still has the capability to jump around. And so this puts the oocyte in a very vulnerable position because now its main goal is to transmit high-quality genetic material to the next generation, but it has to deal with these jumping genes that are creating DNA damage. Previous work in the Bortvin lab has examined a role for line 1 retrotransposon activation in mouse fetal oocytes, and they were able to correlate the amount of line 1 in oocytes with increasing amounts of DNA damage and decreasing cell viability. So in other words, oocytes that express highest amounts of line 1 retrotransposon are those that are preferentially eliminated from the oocyte supply to prevent their propagation to the next generation. And in some cases, up to 80% of oocytes are eliminated during this window of line 1 activation. And this is really significant because female mammals are born with a finite supply of oocytes. So in our recent study, we wanted to further implicate line 1 in oocyte survival during this window where we see massive oocyte attrition. And so to do this, we wanted to identify ways that line 1 is toxic to oocytes. And by doing this, we looked at the activity of line 1 and tried to inhibit this. So first what we did was inhibit the replication ability of line 1, and the mechanism that line 1 uses as a retrotransposon is similar to that of retroviruses. So we were able to block this using a drug that's commonly used to treat HIV infection called AZT. And this, using genome-wide analyses, we saw actually prevented activation of a branch of the innate immune system that we suggest is involved in eliminating oocytes in this way. Line 1 also creates DNA damage independent of its replication activity. And so to account for this, we also mutated the DNA damage response by mutating checkpoint kinase 2. And by mutating CHEK2, we effectively made oocytes ignore the fact that they have a lot of DNA damage. So line 1 can move around and it won't notice. And so by combining these two perturbations, we were able to prevent oocyte death almost completely by the time the mouse was born. And so you can see this here. In the photo, in white is a control ovary that has physiological amounts of line 1 expression that is not perturbed. And then on the right, in purple, you can see that this is a case where line 1 activity has been evaded because we've treated with AZT and mutated checkpoint kinase 2. So by the time the mouse is born, we've now increased the oocyte supply greatly. So now that we have a system to preserve these oocytes that would normally be killed due to line 1 toxicity, we wanted to take a closer look at the quality of these cells. So to do this, we looked at the amount of DNA damage and line 1 expression in oocytes upon evading death. And what we saw was that initially there was more DNA damage in these cells, suggesting that they were the lower quality that would have been eliminated. However, a few days later, after birth, we looked again, and they were able to repair this DNA damage and reduce line 1 activity, suggesting that they're able to overcome this initial heterogeneity in quality. And so we then went on to test the developmental and functional capacity of these oocytes and found that they acted comparable to that of the control. And so we suggest that now we can increase the oocyte supply in mice and not have too many obvious functional consequences. So this seemingly neutral effect of preventing death of oocytes with high line 1 activity raises a lot of exciting questions. First is thinking about the offspring that arise from these oocytes that were initially exposed to high line 1 activity, and whether these offspring are going to be more predisposed to disease as adults or have more mutations. On the other hand, maybe having this line 1 activation and preserving this window of line 1 activity is important on an evolutionary timescale, because by not having this ability to test new rearrangements of the genome, perhaps we won't be able to adapt to stress as well.